Our speaker for today is Professor Shawn Chen. He is Nasrat Muzain Chair Professor in Medicine and Technology at the National University of Singapore. He is the founding editor of the journal Theranostics. He is also the inaugural editor-in-chief of ACS Nano Medicine. He was elected as AIMBE Fellow in 2017, SNMMI Fellow in 2020, member of European Academy of Sciences in 2024, member of Academia Europaea MAE in 2024, and member of Singapore National Academy of Science SNAS in 2024 again. Received NUS School of Medicine Best Researcher of the Year in 2025, JBN Trailblazer Award in 2023, SNMMI Michael J. Welsh Award in 2019, ACS Bioconjugate Chemistry Lecturer Award in 2016, among many other awards. He became a member of the Advanced Materials Hall of Fame in 2023. He is also the past president of the Radiopharmaceutical Science Council, Society of Nuclear Medicine and Molecular Imaging. His research is largely focused on the development of various forms of theranostics, that is, combination of diagnostics and therapeutics, for example, radiotheranostics, nanotheranostics, immunotheranostics, magnetotheranostics, phototheranostics, etc., that can be clinically translatable. Thank you so much, Professor Chen, to have joined here. Please, you can proceed with your lecture now.
Thank you. See. All right. It's really my pleasure and privilege to be able to present some of my work at this ACS Science webinar. I'm a little bit under weather today, coughing, and I have a cold, so bear with me. Title of my talk is Cancer Theranostics. As Niha already mentioned, I have several disclosures. And I also want to promote the journal, the new journal, ACS Nano Medicine. It has already started to accept new manuscripts from June 3rd. It's been over almost 3 months. And we received good number of submissions. And our first 5 or 6 papers will be online in a week or 2. And for this new journal, if you're not familiar, we have some new features for this journal. In particular, we have opinion leaders to be interviewed for each issue. Our very first issue, we have Prof. Bob Langer from MIT, who's the legendary nanoscientist, has the highest H-index, and he's the founder of Moderna. Moderna, of course, had the mRNA vaccine that saved billions of lives. So we had the honor to interview him, and that interview piece will also be online later this month. We encourage you to submit your papers, whether it's a review article or original paper, to ACS Nano Medicine. We also are looking for early career board members, ECB members, those who are junior PIs with website, willing to publish in the ACS Nano Medicine, especially original articles, and be an active reviewer for the journal. Then you are eligible to be a ECB member of this ACS Nano Medicine, right? All right. Coming back to the topic, theranostics, it's coined from 2 words, therapy and diagnostics. It's called theranostics. Sometimes it's called theragnostics. The purpose of theranostics is to develop molecular diagnostic tests and targeted therapeutics with the goals of individualized treatment by targeted therapy to an individual's specific disease subtype and the genetic profile. Usually, it's diagnosis followed by therapy when the patient goes to clinic. He or she will be diagnosed for whatever disease, then a treatment plan was applied. Especially for cancer patients, it's important to stratify patients who are likely to benefit from a given treatment. There are many options, and which option would be the best for a particular patient is very critical. Also, during treatment, you need diagnosis, whether it's in vitro diagnostics or in vivo imaging, to monitor early response to the treatment and then predict treatment efficacy. Oftentimes, diagnostics and the therapeutics are co-developed. For ultrasound, for example, it's so-called sonotheranostics. For immunotherapy, you have immunotheranostics. For MR or radiology, you have magnetotheranostics. For optical, you have optotheranostics or phototheranostics. Then for nuclear medicine, you have radiotheranostics, or radiation oncology, you have external beam radiotheranostics, and so on. We also have a book, Cancer Theranostics, published 11 years ago, highlighting different forms of cancer theranostics. 11 years later, this summer, I have this book re-edited. The second edition has already come out. You can buy it from Amazon or from the publisher website. For nanotheranostics, because we only have 35-40 minutes, so I want to only focus on one topic, nanotheranostics or nanomedicine in general, cancer nanomedicine. We have a nanomedicine TRP translational research program here at the National University of Singapore. Thinking about nanotheranostics, basically, you have a nano formula, nanoparticles, whether it's inorganic materials, organic or polymeric materials, both as diagnostics and as therapeutics. Sometimes they are put in together. Oftentimes, you label a nanoparticle with different forms. It could be radioisotope, could be a fluorescent dye, quantum dots, magnetic nanoparticles, or ultrasound bubbles, and so on, to allow to measure the distribution and the behavior of these nanoparticles. Right? Many of such nanoformulas, whether they are PLGA nanoparticles like bind 014 or some sRNA nanoformulas, or sometimes inorganic nanoparticles such as CDOTs, silicon nanoparticles have been radio-labeled or fluorescently labeled, put into human to understand the biodistribution and the redistribution and the behavior of the nanoparticles within the body. Right? For tumor registration, patient stratification, therapy regimen selection, and the therapy response monitoring all require some sort of diagnostics, whether it's IVD, such as the sensitive biosensor and nanosensor measurement, or in vivo whole body readout. Can be very important to understand better how nanoparticles behave. Yet, we have a lot of publications in many different journals. Only a handful have been approved by FDA. There's apparently a disconnect between the large body of literature and the small number of clinically viable nanoformulas. So what is going on here? Is there an opportunity for nanotechnology to play a key role in cancer detection and the diagnosis of the part of the theranostics? How can we improve nanoparticle delivery? What are the advantages of nanotheranostics over conventional drugs, whether it's biologics, proteins, peptides, small molecules, or conjugates, and so on? And how can we better perform preclinical tests of nanotechnologies to ensure the efficacy and the safety and the smooth translation into clinic? There definitely a lot to be thought about in understanding better the tumor biology will help immensely to deal with the challenges and the opportunities in cancer nanomedicine. Plus, nanoparticles formulas are often more complex than other structures, right? So machine learning and AI can be very critical based on the a priori or prior art of different nanoformulas. The based on the whether it's for detection, in vitro diagnostics, whether for imaging and for therapy, there are many different forms, and those nanoparticles have different compositions, structures, shapes, aggregates, surface chemistry, and then protein corona and so on. All the inputs can be critical to understand better based on the proper models and have the good output and high throughput screening of these nanoformulas for different purposes. So it does provide a set of tools capable of performing time-consuming and the result perception tasks, offering unique opportunities for nanotheranostics. And also, we can predict the next generation nanotheranostics with reliable data sets we already have and advance the machine learning models to offer better clinical benefits to the patients eventually. So we started with something rather simple with the hope of translating nano formulas into clinic. One way out is the simplicity. Nanoparticles are complex. So the simplest nanoformula we can think of is single component. Itself is both API and excipient. So what we did here, we have a chemotherapeutic drug, CPT, camptothecin, conjugated with the albumin binder, MS blue dye or other albumin binder. This way, it's an amphiphilic structure. Yet you have the prodrug, the camptothecin derivative here. It is indeed both API and excipient, self-assembled into micellar structure, nanoparticle, 80 nanometers in size. Because it's amphiphilic, it can also load other chemotherapeutic drugs like paclitaxel into this structure. Then it forms a vesicular structure. It can load very efficiently 1-to-1 mass ratio, which is very significant. Conventional liposomal or albumin-based vehicles to load peptide. It's somewhere between 5 to 10%. Here it's 100%. It's very, very effective. So when you have this formula, you have a combination of therapy, paclitaxel, one chemotherapy, and then camptothecin, another chemotherapy. So the combination has some additive effect in the nano formula, shows better efficacy than the already approved FDA formula like Abraxane and Onivite. Right? We can also radio label such nano formula with a chelator, then radioisotope for imaging and for therapy. We label or conjugate DOTA chelator onto this nanoparticle. It can radio label copper 64 for PET imaging and the lutetium-177 for beta therapy. Because you have both chemotherapy, camptothecin part, and also radionuclide therapy, lutetium-177 part. You can titrate the dose of radionuclide and a dose of the chemotherapy. So you can have high dose of radionuclide, low dose of chemotherapy, or high dose, say here, 1 millicurie of lutetium, and a relatively low dose of camptothecin. In both cases, you see almost complete tumor eradication in xenograft models. This shows very high efficacy in preclinical animal models, in the artificial subcutaneous tumor models. So we pushed very hard, radio labeled the same nanoparticle, and put that into human. for first-in-human study. This is the IIT investigator-initiated trial IIT study. We labeled this nanoparticle with gallium-68, another PET isotope. You see here whole body distribution in the cancer patient. From 5 minutes up to 4 hours, we were able to get the projection images of the patient over time. Here are 5 snapshots of those. Initially, you see a lot of activities in the blood or the ventricular system, the heart and the ventricles, and the other major blood vessels, and of course a lot in the liver, like many other nanoparticles. Over time, there's clearance from the body and more into the kidneys because those micellar structures collapse. So there are particles into the kidneys, and then of course into degraded forms, not nanoparticles anymore. They get cleared out of the body. Everything looks fine, like many other different nanoparticles. The only caveat here, the tumor uptake is not high enough. Even though PET-CT does show good contrast of the tumor, this is a colorectal cancer patient with the primary tumor identifiable from PET-CT, but the absolute tumor uptake or the standard uptake value, SUV, is not high enough to warrant further clinical application for treatment of patients. So here, this nanoparticle with EPR effect, very nice EPR effect, enhanced permeability and retention in patients, in animal models, did not seem to work very well in patients. So the conclusion of this ACS Nano paper, published earlier this year, we basically concluded that it's not very positive. It's conventional wisdom of using nanocarrier to maximize chemo drug delivery through EPR effect or passive targeting is fundamentally defective. That conclusion is not trivial. We actually concluded this based on our discrepancy between animal studies and the human trial to show that EPR effect, if any, in the human situation is not as significant as in preclinical mouse models, right? So that comes to the previous soul sorting questions. What's wrong with our study? What's disconnect between preclinical and clinical studies, right? Oftentimes, even though we don't have as much clinical data, but clinical studies definitely not always reflect what we observe preclinically, right? So what do we do? There are a lot of features nanoparticles have. We should take advantage of nano. We don't want to have hyper. I don't want to have hypo about nanosystems. We have to understand better how the nanoparticles behave in the body, right? Based on the morphology, aggregation, surface charge, size, and the composition, right? Whatever nanoparticle is injected through whatever route, subcutaneous, intravenous, intranasal, or local administration, our body would always have a defense. Our body will defend against any nanoparticles as foreign invader and maintain its homeostasis. Right. So any disruption or dysregulation of the immune function by nanomaterials could have significant implications for human health. Right. For cancer, cancer is an immune disease, it's a chronic disease. Immunotherapy has been very successful in combating cancer. Either it's cytokine, vaccine, cell therapy, CAR T, or other ACT adoptive cell therapy, checkpoint inhibitors, antigen blockade, all those immunotherapies. If we apply the nano component into this, we might come up with immunotheranostics that are to our advantage, because nanoparticles will have some body immune response. That immune response, if that's going to change the tumor microenvironment, stimulate the immunosuppressive microenvironment into an immunoactive environment, then these nanoparticles not only behave like a carrier, but also have its own function to change or engineer the tumor. So that way, we might find a better treatment. Not only what we have listed, 5 different types of immunotherapies, but also with the nano regulation of the TME and have better immunogenic cell death or checkpoint blocker. So also we have to deal with cancer metastasis, not only the primary tumor, because most cancer patients die of metastasis, not from the primary tumor. Then a lot of different ways we can formulate immunotheranostics or therapeutics through different ways in combination with other therapy, phototherapy, radiation therapy, chemotherapy, and so on. So that's why a lot of nanoformulas become very important. So we want to only emphasize one topic today. It's about mRNA vaccine. Cancer vaccine has been very, in many different forms. mRNA cancer vaccine has been at the forefront recently, especially with the advancement in mRNA vaccine against COVID-19. And a lot of principles, ideas learned from that have been applied to cancer as a prophylactic as well as therapeutic. For mRNA, it's not only good for immunotherapy as mRNA vaccine, for example, also it's opposite to siRNA. It's good for protein replacement, genome editing like CRISPR-Cas9, and the cellular reprogramming, whether it's ex vivo or in vivo CAR T therapy or other cell therapy. So for any of this, we require delivery of mRNA. Naked mRNA does not work. We have to have a good way to load it. First step is tissue or cell-specific accumulation, then endocytosis, or internalize the mRNA nanoformula to intracellular domain. Then in the endosome, in particular, we want to have rapid escape. Otherwise, everything gets chopped there. So they can do a membrane rupture, fusion, destabilization, and a pore formation to allow endosomal escape. Of course, there are also other ways. You don't allow the particles to go to endosome, bypass endosome. That's another option. Then you have to release mRNA, whether it's passively or it's actively through pH dependence, enzyme, or redox. Then you have to translate the mRNA into protein efficiently, right? Every step of the way through these 5 steps can be critical in the outcome of mRNA delivery. As a carrier, it can be viral or can be non-viral. For non-viral carriers, most successful one is lipid-based. For lipid-based nano formulas or LNPs, oftentimes it has 4 components: helper lipid, ionizable lipid, cholesterol, and pegolipid. And sometimes can be the 5th component, have the targeting moiety or targeting ligand for different cells, immune cells, tumor cells, or macrophages, and so on. Those can be made in a very uniform particles through a microfluidic device. There are also polymer-based and biomembrane-based carriers, all 3 different forms. Polymers, in principle or ideally, can replace the 4-component lipid nanoparticles. Theoretically, it allows endless synthetic capability, versatile structure, and a robust stability. can be a single component to have all the functions of the four-component lipids. That's why people have spent a lot of time trying to engineer different polymer structures to allow efficient delivery, because polymers have been successful in delivery siRNA, DNA, and other forms of nucleic acids. Right? For mRNA, it should also work to some extent. Here's one example. We use a simple copolymer, PHTA polymer, hydroxyl tertiary amine repeating units. It has tertiary amine, it has hydrophilic backbone, we see hydroxyl groups, and hydrophobic side chains, especially fluorinated side chains. This polymer trying to replace lipids, especially when lipid nanoparticles with PEG lipid at a high concentration for tumor applications. The PEG lipid might have inflammatory response. It can be highly inflammatory, could be detrimental to the patients. That's why we want to have polymer as an alternative to the lipid nanoparticles. Here, this C18 PHTA polymers, amphiphiles, achieved successful mRNA delivery migration from the injection site, subcutaneous injection through lymphatic vessels to the lymph nodes, and then recognize dendritic cells, and then translate. Here we use a model mRNA, firefly luciferase F-look mRNA in the lymph nodes, and then use the ova to treat xenograft tumors. This one, although kind of works, did not have chance to move forward to the next step, because this polymer, even though it's relatively simple, it's not biodegradable. So we eventually gave it up, thinking it may not be good as the API, the excipient to carry mRNA. In the end, when we think about GLP toxicity, IND enabling studies, you may not be able to get a DMF drug master file as a excipient or carrier for mRNA, even though in principle it works. So we moved on to try different formulas. Yeah, in the previous case, polymer kind of works, not to say it's not biodegradable, and it's not much better, not superior to lipid nanoparticles. So we thought, what if we combine the advantages of polymer and the advantages of lipid nanoparticles? So here's the one example we published last year. We have the preformed lipid nanoparticles, four components. Then on the surface, we wrap around a polymer. This polymer has some function. It serves two purposes. One is to shrink the particle a little bit, make the nanoparticles more compact and more stable, enhance cellular uptake. At the same time, this polymer with tertiary amine behaves like a STING agonist. So this way, we have the combination of STING agonism and mRNA delivery. It works both as prophylactic, or means you vaccinate the mice before tumor inoculation, as well as therapeutic. That means you inoculate the tumor first when the tumors are established, then you subcutaneously vaccinate the mice for multiple times. You do see a good delay of tumor growth. This way, we have five components. Talking about translation, we also face the challenges, right? Because it becomes more complicated. That polymer also does not have, it's not the drug master file or the IND available. So that's why in the end, we came back, rethought about lipid nanoparticles. right? The four components, as I mentioned earlier, you have cholesterol, DSPC, ionizable lipid, and the PEG lipid. People work a lot on ionizable lipids, try to change that structure, allow different behaviors. Less has been done on PEG lipid. Right? People have tried to replace linear PEG with different molecular weights, with a branched PEG, with a circular PEG, and so on. So what we did here is to replace PEG with a different structure. We used the albumin binder. Of course, we can try many other different molecules as an alternative to PEG. So this albumin binding MS blue dye, or actually you can have a library of many different forms of albumin binders. It could be lipids, could be other dyes, molecules, and then we pick the MS blue AB19 in this library. It serves at least two purposes. Now we purposely have a protein corona, albumin corona on the surface, because you have a bunch of MS blue dyes on the surface of these lipid nanoparticles, and each MS blue can bind one albumin protein. You now have a tight albumin corona on the surface. Also, the albumin binds to albumin receptor or neonatal receptor on the surface of dendritic cells. It allows active targeting of dendritic cells, right? So it's different from pegylated LNPs, which may have a non-specific protein corona, different proteins on the surface. Even though it has PEG, still some proteins will bind to the surface. Does not have specific binding. So here we have imaging as a tool to show the different behaviors of PEG lipid versus EB lipid. Using optical imaging after subcutaneous injection, the conventional PEG lipid nanoparticles fall apart readily and leak from a lymphatic system or from an injection site to the circular system, to the blood, into the blood vessel. Then you have locked into the liver, spleen, and other major organs. If the EB lipid nanoparticles, it's more stable because the protein corona, it has little leakage from injection site to the other organs, tissues, but the more effective into the lymph nodes. You see from the ex vivo imaging particularly shows very manifold increase in lymph node delivery. To show better, we used PET imaging in large animals, in mini pigs. Here, PEG lipid formula versus EB lipid formula after injection, arrows pointing at injection site, and the PEG lipid is a lot more throughout the body. The EB LNPs is much less throughout the body, but a lot more into the lymph node here, especially at the groin in pig, the hind limb injection, you see a lot migrate into the groins. It's very effective. And also microscopically through EM, and we use the model nanoparticle, use silica nanoparticles. This way we do see much better delivery for the EB nanoparticles compared to PEG nanoparticles, how much more goes to the lymph nodes than the conventional PEG lipids. Of course, then applied to different models, tumor models, for example, B16 melanoma, HPV cervical cancer, and HPV oral cancer all showed better effect through this EB vaccine. And the influenza, H1N1 influenza, and the COVID-19 animal models, they all show better efficacy due to better delivery and less leakage into circulatory system. So it's more effective and more safer for this EB formula. That's why we hope to have this particular structure to move on into the next level. And finally, we also work on cancer stem cells. As cancer vaccine oftentimes target immune cells or chemotherapeutics with target tumor cells, but there's a small portion of cells in the tumor region, the self-renewing cancer stem cells or stem-like cells, CSCs, this subgroup of tumor cells within tumor mass underlie cancer initiation and evolution. It mediates cancer progression and recurrence. They are resistant to conventional therapeutics because they are more quiescent, right? How to track and eradicate those residual cancer stem cells after debulking of the primary tumor can be very critical. There have been cancer stem cell vaccines, and it's very tedious. How that's been done? You have to debulk the tumor, isolate tumor stem cells or cancer stem cells, and those CSCs being destroyed or lysed into CSC lysate. Then inject back the CSC lysate to activate the immune response. The efficacy is limited, and it takes a long time to have the vaccine ready. Oftentimes not sufficient because you don't have enough CSCs to make sufficient amount of CSC lysate. If we use CSA-specific antigens, CSAs, shared by different types of tumors, one such example is ALDH1A1, and we might be able to develop CSA targeting vaccine not using the tumor lysate, right? Use autologous tumor lysate. So here we actually developed a nano vaccine target both cancer stem cells and the bulk cancer cells for post-operative cancer immunotherapy. So this design is a little bit convoluted. So first, we use ALDH1A1, that's one of CSC antigen CSA. Overexpressing tumor cells, we isolate the cell vesicles. Then you encapsulate the different antigens into the nanoparticles, especially tumor-associated antigens, and also on the surface of this CSA or CSA membrane vesicle, you have a dendritic cell targeting aptamers. We choose a particular aptamer targeting DC cells. In that nanoparticle or nanoformula, we also have epigenetic regulation for lysosome proteins. It's very critical because a lot of times the antigens, even though expressed, do not cross-represent into the surface. These lysosomal proteases can degrade those antigens very quickly. So we now deliver the particular protein, YTHDF1, into dendritic cells, try to restrict the translation of these lysosomal genes to inhibit such lysosomal proteases that slow down or prevent the degradation of the internalized antigens in the DC cells. So this way, we have delivery of, here this scheme again, delivery both TAAs and the CSAs into dendritic cells. And also we have this particular inhibition of this lysosomal protease inhibitor. So This way, it has better or improved antigen cross-presentation, enhanced cytosolic export, and then a good presentation of antigen on the surface. So in the end, we have a good immune response, this so-called nicer nanovaccine, and the inhibition of post-surgical tumor relapse. I did not provide enough details, but then we can discuss offline if anyone has any specific question. All right, to quickly conclude, to better understand the role of tumor microenvironment in the tumor development, progression, and metastasis can provide us a lot of new opportunities to develop new cancer theranostics. Using nanomedicine to deliver different therapeutic molecules into the tumor microenvironment instead of directly into tumor cells, especially for the reversal of the immunosuppressive TME into immunostimulating TME, can offer new options to modulate the tumor and treat tumors more effectively instead of conventional wisdom of maximize the delivery of chemotherapeutics to tumor cells. mRNA delivery to the cancer site or to the lymph nodes or to other immune organs for effective cancer immunotherapy requires lipid polymer or hybrid nanoparticles that are DC specific or other immune cell specific, can have efficient protein expression. Also, vaccine alone is not going to work like in most cases. We need combination, combining mRNA vaccine and other therapeutics such as chemo, radio, nuclear, and immunotherapy will be exciting new frontiers in cancer management. With this, I'd like to thank my collaborators and funding agencies and my lab. Thank you.